this new self-amplifying RNA LNP.
Self-reproducing, like the
same.
Okay.
It's to launch it.
Jessica Rose, you all know her, and if you don't, Jess, we're not going to do the thorough 30. We're going to do like a 70,000 foot overview. You've been on recently. We were talking about DNA fragments found in COVID jabs. Your title, and that is all. What is it again? I keep forgetting. It's a statistical biologist, analyst, PhD.
We'll call me a computational biologist with an immunology background. How about that?
That's damn good. Computational biologist with an immunology background. That is it. Okay, so you put out some posts last week. I see them because I follow you and they actually do appear on my feed. I don't even understand what I'm reading in order to even attempt to summarize it in a retweet. So I said, you'll come on and talk about what the hell is going on. Tell us what's going on.
Recently, I can't remember if I told you this, but I went to Japan with a bunch of people, the International Crisis Summit people, and we went there with the intention to advise some parliamentarians whose ears we had about the dangers of this new self-amplifying RNA LNP technology. So the reason why we went there to do that was because they were about to launch it and like actual into the public as a product, a COVID product into the elderly population. They since have done that. We did make some noise. We did save probably some people from getting injected with this crap, but they did launch it. The good news is not very many people actually said, yes, I will agree to being injected with this stuff. So maybe we made more of a difference than I think. But here's the deal. This is using the same lipid nanoparticle technology as these modified mRNA LNP technology COVID shots, the Pfizer and the Moderna. So as you guys already know, this involves the genetic material for the spike protein being encased in this lipid nanoparticle, injected into you, distributed around your body, and your cells and their machinery are using this genetic material as a template to produce the spike protein in your cells. So it's more like a prodrug. Your body is needed to produce the byproduct of the genetic material. So lots of problems with these products, but that's not what this is. This is different in a fundamental way. So the reason why it's called self-amplifying is because it's self-amplifying. The way that it's self-amplifying is that there's this virus type called an alpha virus. This is in the, what is it called? The toga virus. Anyway, it's in a specific family of viruses that is unique in that it carries a gene called the RDRP gene, which is the RNA-dependent RNA polymerase, which means it has a function such that it can photocopy the genetic material of the virus as part of the way that the virus optimizes its survival. It's one of these techniques that viruses have evolved, these specific types, to ensure their own survival. One of the types is called Venezuelan equine encephalitis virus. Equine is horse, Venezuelan is Venezuela, encephalitis is inflammation of the brain. This is bad. This is one of the viral genomes that they use in the biotech realm to manufacture these self-amplifying products.
What would be the difference between self-amplifying and self-reproducing? Like the
same.
Okay.
For all intents and purposes, it's the same. It's in reference to the genetic material. So it's like a little enzyme that photocopies the genetic material that will be translated into protein eventually. So it just, the fact that it carries its own, it just ensures that there's more copies of the template. They've taken this backbone of this genome of some alpha virus. They've kept in this gene that can photocopy genetic material. They've taken out the guts of whatever virus it is, like the subgenomic stuff, which is the stuff that is responsible for building the virus, like the glycoproteins on the outside, the capsid, all the viral guts that's necessary to make an entire viral particle. And they've substituted in the case of the product they put into the Japanese population, the spike gene. So effectively, what this does is it's the same, the LNP thing is the same. You inject it into the muscle, traffics all over your body, dumps this genetic material into your cell, but instead of this mRNA that was the genetic material in the modified mRNA shots being translated into protein. First, you have this RDRP thing being the photocopying machine of the genetic material, which is subsequently translated into protein. So what you effectively have is a nonstop production of an amplification of the genetic material for nonstop production of spike protein. The manufacturers of these horrid things claim that this is awesome because you don't need as high a dose initially. You don't need 30 micrograms or 100 micrograms in terms of Pfizer and Moderna, respectively. You only need 0.1 micrograms. But the thing is, it doesn't matter because this stuff is going to be making its own stuff, which is going to be translated into protein ad infinitum. Total metabolic expense of the cell.
Okay, hold on. Let me see if I can back this up and simplify this a little bit. The substance itself is reproducing or not reproducing, photocopying on its own once in the subject that is the
cell.
What is it reproducing from? Because I mean, presumably it's not splitting, so it's acquiring. And is it converting or is it creating matter in order to amplify itself?
It produces the string of mRNA that will be used for translation by converting to, I think it's CD. Anyway, there's a process that it goes through in order to make the copies of this mRNA. So it makes repeat copies of the mRNA from another template through a process. I could describe what it is, but it's just going to get more confusing. So it's safe to say that from the template, you put a piece of paper into the photocopier machine and it spits out copies of that paper, right? That's what this RDRP thing is doing. And that piece of paper is the mRNA. It's just, it has this ability through, like the way that these things happen to keep copying this. And actually, it's really important for people to understand the analogy with the photocopier because of the anticipated problem with this that I've been talking about endlessly. Besides the fact that this is going to be really detrimental to the cell, and also the fact that this is stemming from an alpha virus template, I have to set a scene here, okay? First of all, I want to tell you something. Besides the fact that they have put this product into people in Japan, on November 11th, which was 6 days ago, the FDA gave the green light for a phase 1 clinical trial to start to have the same self-amplifying RNA LNP technology tested in the context of the H5N1 virus, which is bird flu virus. So instead of the spike genes being spiked into this template, they have these genes, probably the hemagglutinin and the neuraminidase, which are these surface proteins from this H5N1 being translated. And of course, it carries this photocopying machine that reproduces the mRNA like an assembly line.
And I'm not trying to be stupid or smart, but or too smart, but like a photocopy machine, it's reproducing it. It pulls a sheet of paper out and then it converts it into something else. Is the amplification process creating? Is it converting something in the body already into the mRNA or just energy and
both?
Okay, I'm going to ask the other stupid question. I know we're going to get there. Cancer is when cells reproduce uncontrollably. This is not reproducing cells per se. When I hear this, I immediately only think of cancer, but only because I don't think I'm understanding this process compared to cancer reproducing cells without control.
So different levels. So the cancer is outgrowth of cells. You're absolutely right. So that's like the bigger level. What we're talking about is inside the cell. So this lipid nanoparticle dumps its genetic material into, just to visualize, one cell. So this photocopying is going on somewhere inside the cell, probably in the cytosol. It's producing tons and tons of copies of the messenger mRNA. The messenger mRNA finds ribosomes somewhere and that subsequently gets translated into protein. So this is happening intracellularly. So that massive amount of mRNA being copied and the protein that's being produced as a byproduct is all accumulating in that cell. The way I'm imagining this is that probably the cell's going to die because all the metabolic expenses that are being used, like it's a lot of work for a cell energetically to produce proteins. And I imagine that the scale, because this is self-amplifying, is many times higher. So the cell's either going to probably give itself a signal to apoptose, which is to self-destruct because, something's wonky, or it's going to pop because there's going to be too much crap in it.
Cancer is the cellular growth itself that grows uncontrollably. They don't.
Yeah.
They don't split.
Well, it's the multiplication of a single cell. So it's like separate cells can become cancerous, and cancerous cells can proliferate, which means, they're double.
The risk of the amplifying mRNA within a cell, you say the cell would either die or pop. No risk that the cell spontaneously grows or reproduce, or I don't know.
I see what you're saying. That's actually an interesting thing. I didn't think about that.
I'm picturing like the cell splitting into two, like an egg that's now twins, and then the twin splits. And I don't know I'm only thinking cancer and all this. I wasn't even thinking like the cellular death.
No, that's interesting, but I dare say that, I don't know. It's something interesting to think about. It's possible that a cancerous cell could also, like, think about this. If you already have a cancer cell that's transfected, and this stuff starts going on in that cancer cell, then because of the nature of a cancer cell,
you're giving steroids to a cancer, like self-amplification to a cancerous cell.
It could be like
okay.
It's a nice science question you just asked, because like it could be like we don't know. If you already have an existing tumor or some cancer cells that are, metastasized, whatever, and this stuff I didn't even think about that.
Let me ask you this: is the only benefit of self-amplification reduced loads? Like, I could see the benefit of self-amplification, sort of like creating an autonomous machine that you don't need to keep servicing. If it's to say, well, instead of going once a year, now you're going to go once every 3 years, or you just get one and it'll last you for 10 years. If that's the only benefit of self-amplification, okay, let me back it up again also here, because mRNA, it's only now been used for the first time with these so-called COVID shots. Prior to that, there was a scientific use for mRNA, or at least a scientific interest in mRNA for other purposes, but had it actually been implemented safely in any product whatsoever in humans?
mRNA?
Yes.
Not that I'm aware of, but I could just be out of the loop on this.